It's my pleasure to introduce today's speaker, Dr. Kara Lynn Davis from Stanford University. She is going to be talking to us on understanding treatment resistance and relapse in ALL through single cell studies. Dr. Davis received her Bachelor of Arts from Pennsylvania State University, right here, in 1996, and Doctor of Osteopathy from Philadelphia College of Osteopathic Medicine in 2000. She completed her pediatric training at Thomas Jefferson University in Philadelphia and her fellowship in oncology hematology at Stanford. She has been instrumental in leading clinical trials. To name a few, one of her trials led to the FDA approval of first cellular therapy for cancer for CAR T cells. She is also a co-investigator on the Stanford Institutional Clinical Trials for CD19, CD22 bispecific CAR, and CD22 CAR for relapsed and refractory leukemia, and GD2 CAR for childhood brain tumors. She is active with the COG in acute ALL committee, and also co-leads the phase 1/2 trial for immune checkpoint inhibition through COG. Dr. Davis is an ALSF Young Investigator Awardee and is also an active member of our review board. Her talk today will focus on research applying single-cell high-parameter technologies to the study of childhood cancers to identify cell populations and prediction of relapse or treatment resistance. In her talk today, she will cover the current understanding of relapse in BALL. She will also review B-cell developmental changes and decision-making and role of transcription factor and metabolic states in relapse to current therapies. So without further delay, Kara, stage is all yours. Thank you.
Thank you so much for that nice introduction. And it's nice to be here with everybody today. I do want to say I so appreciate the support of Alex's Lemonade Stand. It was my first grant that was ever awarded, and really helped me, continue to do research actually, or get on my research path. So lots of fondness for Alex's Lemonade Stand. And one clarification, I participated in the CD19 Ileana trial as an institutional investigator. Certainly, that was an honor to be part of that, but that all was led through Children's of Philadelphia and Novartis. So today I'm excited to share with you my research and the work that we do here in the lab. I have a couple disclosures, and then we'll jump in. I think most people on this call most likely know that B-cell ALL is really the poster child for success in cancer treatment. These survival curves, which are relatively recent, either from the St. Jude studies on the left or the St. Jude and other cooperative group trials on the right, have really showed this incredible progress that I think is a wonderful example of how the collaborative nature of childhood oncologists and pediatric cancer researchers has led to improvement in cure rates for kids with ALL, which is, of course, is the most common type of cancer in children. But I think for any of us who treat kids with ALL know that relapse is still a leading cause of death from cancer in children. And in general, relapse across cancers is, for the most part, the thing that causes mortality. And so it's still something that we need to learn more about. What are the kind of cells that cause relapse? These survival curves here typify and these are sort of earlier and later studies that have looked at, what is the outcome for children with ALL relapse as we've gone through either successive periods of time where perhaps you would expect that there's new treatments, or even how many relapses patients have. And I think the real take-home message is that, we still have a lot of work to do as these curves are hovering around 50% or less. And so this is, a major limitation for the patients that we see in giving them long-term cure. And even with new therapies, and this is the survival curves and the event-free survival curves from the Ileana trial of the CD19 CAR Kymriah, when it was in the multi-center trial. We still have work to do, as we can see. I think you guys can see my pointer. That the event-free survival here still hovers around 50%. And so again, if you're taking care of these patients, that for some patients, these therapies work wonderfully and can actually be definitive. And for some patients, their leukemia keeps coming back. So this is really the goal of my research program, is to understand and prevent relapse in childhood leukemia and childhood cancer, because I think a lot of these techniques and ideas are applicable outside of liquid tumors, although the actual approach is a little bit different. And so we know that when patients are diagnosed and they come to us, they have billions of cancer cells. We give them treatment, and in some cases, we can monitor their response to treatment by minimal residual disease testing. And some percentage of children, and in this case in ALL, the vast majority will be cured, but there's still going to be a significant portion of kids who relapse. And the way that we think about this and approach this problem is by using single cell tools to be able to look at the cancer, especially at the time of diagnosis, or even can be at relapse time points, and understand and try to take apart the total of the tumor using single cell studies to be able to find cell populations associated with relapse or treatment failure. This little cartoon shows a cytoph, or a mass cytometer, which is sort of our workhorse instrument in the lab, but we also integrate other newer sequencing approaches that are single cell, which, as we know, are also really becoming a lot more accessible over the last few years. And so I think about this a lot like this with cancer and in leukemia in specific, that we know, like in a colony of bees, that the queen is the necessary species to be there in order to nucleate and to make the entire colony happen. Without the queen, the whole thing dies. And so, and you can tell the difference between a queen or a worker or a drone if you can get in and look at them very carefully and notice the differences in how they look. But if you look at a beehive or a colony, this may be what you see, as the outside look. So it can be very hard to identify, where is the queen. And I think about this a lot in the same context with cancer, that I want to know what are the cells that are the ones that are capable of recapitulating the entire tumor, and what's unique about them compared to the rest of sort of the worker bees, so to speak, which we think tend to be sensitive to the treatments that we already have. And when I embarked on this, and this is the first data I ever generated when I was in Gary Nolan's lab as a postdoc, using cytoph, and this was some samples we got from Charles Mulligan at St. Jude. We found that heterogeneity was really a huge challenge in single cell data analysis in cancer. These are 9 patients who have Ph positive ALL. They all would be treated the same way. And when we look sort of in this part of this is a phenograph map. Basically, we see that every leukemia clusters with itself. These are all colored by patient. And so, there's a little bit of overlap in terms of some populations that are shared between patients, but for the most part, the patients are just sort of with each other and separate from the healthy cells, which sort of sit down here. And so I was hoping, kind of naively, that we would use these techniques, do some clustering, and call out populations that are common across patients, and that then we could, interrogate for their association with relapse. So what we did in order to address this heterogeneity was went back to not a particularly novel concept, but something that was published, many years ago by Mel Greaves, and I think a way that we conceptualize often, these different types of leukemia is using the scaffold of normal development as a way to organize this heterogeneity. And so when I was in Gary's lab, one of the first things that I worked on that was sort of a side project to my leukemia questions was using mass cytometry, or cytoph, if we could define the trajectory of normal developing B cells. And we did that in a paper that we published now almost 10 years ago, where we took healthy human bone marrow from 10 different donors and looked at the expression of surface proteins, intracellular proteins, which included transcription factors and enzymes, as well as signaling checkpoints and rearrangement of their VDJ, to be able to sort of line up these cells developmentally from the time that they're going from early lymphoid progenitors to immature B cells that are ready to leave the marrow. And so, armed with that knowledge, as we started to approach leukemia samples and patients, we used that to come up with what we called a developmental classifier for B-cell ALL. I think this is an approach that people use very commonly now when they're doing single cell studies, is trying to sort of take cancer cell populations and relate them to their normal or healthy cognate cell populations. So the way that ours worked is that we would just manually gate based on what we knew from those prior studies, healthy bone marrow controls based on their progression of marker expression into different populations. In this case, we used 15 different populations. And in that 15 populations, we would look at the expression of this 11 protein, essentially signature. And then for every leukemia cell, we would say, which one of these populations does your 11 protein signature most closely match? And then we could assign a developmental population to every leukemia cell that we looked at. And that became a really useful way for us to be able to, look at the data and feel like we could compare populations of cells across patients in a relatively fair way, because we were always sort of normalizing them to their healthy populations. So the first story that I wanted to tell here is something we've been working on in the lab about antigen escape after CAR T cells. So we really wanted to understand if we could find the cells at risk for antigen loss before we gave someone a CD19 CAR T cell. And a little background, of course, on CAR T cells, which I'm sure this audience also knows, is that, there's three main modes of relapse that happen after CAR T cells. And in particular, we're talking about CD19 targeted CAR T cells. The first is lack of persistence of the CAR. And we think that this may be mediated by immune rejection or exhaustion. But this leads to a CD19 positive relapse. So the cells still express their target, and we think it's a kind of a T cell problem. You can also have CD19 negative relapse, where now the cells have lost that target on the surface of the cells, and there have been, and I'll touch on the different mechanisms that we think are part of that process. And then the third is lineage switch. So when the leukemia goes from looking like a B cell to looking like a myeloid cell, or even sometimes a T cell, there's a lot of heterogeneity there that has to do with most likely plasticity. So what we do know, though, of course, is that overall survival after a relapse after CAR, of course, is even worse than sort of just the survival curves that I showed for relapse after chemotherapy. And of course, these are all patients coming in who most likely either have had high risk disease or they've already relapsed, to begin with before they came to CAR. So as I mentioned, they're now with these CD19 targeted therapies in leukemia and lymphoma, there's been a lot of work to understand, the mechanisms of CD19 loss. Of course, you can recognize frameshift mutations in the CD19 gene itself that leads to loss, transdifferentiation to myeloid cells, which I mentioned as a lineage switch. You can have disruption of membrane trafficking so that the CD19 basically gets stuck inside the cell and can't make it to the surface. So then, of course, the CAR T cell doesn't see it. And then splicing mutations can happen where basically the CD19 mRNA is misspliced, and that leads to a protein that either is not able to be localized to the surface or perhaps lacks the epitope that the CAR would actually bind to. And then finally, you can have basically silencing of the CD19 promoter as a mechanism. And of course, a lot, more and more is coming out about this as we're studying these and as we have more cases of this across the different malignancies. So we wanted to understand, this process in ALL, and we went into this question with two possible hypotheses. So the first here on the top is sort of exemplified by a paper that was published by Catherine Roberts and Charles Mulligan and Yakwe Zhao from St. Jude, where they looked at actually blinatumomab response. And the idea here is that, your cells are uniformly CD19 positive before you get CAR, but then after CAR, there's some cells here that have the capability of losing 19, and the pressure of the CAR or the blinatumomab kind of forces them to lose 19. The other idea would be that you actually have sort of hidden or infrequent CD19 negative or low cells that pre-exist the CD19 therapy, and these cells are capable of growing out and recapitulating a CD19 negative relapse. So we started out with a really exceptional cohort of patient samples. These were given to us, in collaboration with Steve Grupp, David Barrett at Children's of Philadelphia, from patients that had been treated on some of the early institutional CD19 trials and had been expanded in mice. So these were all actually PDX expansion samples. And we had in total about 45 samples, but we actually had a nice cohort just at the pre-CAR time point, so before the patient ever saw a CD19 CAR. And we had patients in kind of every group that we could look at. So we had 6 patients who were going to have a complete response. We had 11 patients who had a CD19 negative relapse. We had 4 patients who had a CD19 positive relapse, and 4 patients who were completely refractory to the CAR. We looked at these with CyTOF, with our kind of what our standard B cell panel is, with a little bit of a few additional markers that we put in specifically here. And I just want to also, highlight here that all this work has been led by Pablo Demisi, who's an instructor in the lab and has really done a remarkable job with this project. And this is all his hard work. So when we looked at the samples using CyTOF before they got their CAR T cells, there was really no significant differences in either the median expression of CD19 before patients got their CAR or the frequency of CD19 positive cells between the different clinical outcome groups. Basically, they all express 19, at relatively the same frequency. But when we used our developmental classifier, and in this case, now we're specifically looking at the pre-CAR19 samples from the patients who were going to have a CD19 negative relapse, and then their paired 19 negative relapse sample. And we could see that there was an expansion in this pro-B2 population, which is pretty typical of where we see ALL cells expand along the B cell developmental trajectory. But what was interesting and was absent in the other subgroups or sort of clinical outcome groups, which I'm not going to show for time, was that let me see if I can get my laser pointer working here. That we saw this expansion of cells in the post-19 negative relapse time point in this population we call early non-B cells. And early non-B cells for us, because we don't have, hundreds of markers included here, are basically lineage negative cells. They don't express any definitive B cell markers like 20 or 19 or even 10. They have some 34, so they look like they're early, but they're not necessarily any particular lineage. So we were interested in this. And so what Pablo did was go back and think about this classifier tool that we use to classify the cells. And of course, one of the proteins that we use to classify is CD19. So we asked the question, what happens if we take CD19 out of the definition of these cells? So like rerun the classification tool, but exclude CD19 from the definition of cells. And we did that. And what we found, I'm not showing it here, is that if you use healthy bone marrow to do this, it doesn't really change the classification of the cells at all. Like plus or minus 19, they pretty much classify the same way, which makes sense in some ways because, in order to define a cell, the co-expression of these proteins is relatively, well coordinated in healthy development. And so just the classification with or without 19 didn't make a big difference. But in the leukemia cells, we actually found that, it helped us better refine what we were seeing. So what we found is that we identified a population we called discordant pro-B cells. So if we included CD19 in their classification, they were called pro-B2 cells. If we classified them without CD19, they were called early non-B cells. So this was sort of the, where those cells were coming from. And this is in contrast to concordant pro-B2 cells, in which if we use 19 or not, the cells stayed in that same category of pro-B2 cells. So there was something different about these discordant cells, and it wasn't just 19. They actually had different levels of expression of additional B cell proteins, which was why they were sort of moving into that early non-B population. So Pablo went back and did the same classification exercise without 19 in all of our patient groups, and we found that these discordant cells are present in all leukemia samples, but not present in healthy bone marrow. So we looked at 8 healthy bone marrows. We couldn't find cells that had this same phenotype, but they were present in de novo ALL samples, sometimes at high frequency. And these were samples that had never seen therapy. They're diagnostic de novo ALL samples that we had from the St. Jude Tissue Bank, again, in collaboration with Charles Mulligan. But certainly we saw them across patients who went into CR, patients who had the 19 negative relapse, one patient who had a positive relapse, and at low levels at patients who were refractory. So we started to focus in on these discordant pro-B cells as a candidate for cells that had the possibility of losing 19. And so Pablo went and just did differential expression of the proteins we measured between the patients in the pre-CAR sample, from the patients who were going to have a complete response to patients who would be CD19 negative. And what we found actually was the transcription factor Ikaros, which we know is associated with relapse in ALL, that we saw less Ikaros protein in these discordant cells from the patients who were going to have a 19 negative relapse compared to patients in CR. And we also looked at PAX5, which we know is a regulator of CD19, and that trended to be also lower, but of course, not statistically significant. And it may play a role, of course, that we just couldn't see yet here because of the numbers of samples that we had. We went actually then and did single cell RNA sequencing with the phenotyping as well, which is AbSeq, similar to SiteSeq, in 9 samples, both in patients who were going to have a CR and in patients who would have a 19 negative relapse. And again, looking at gene expression after clustering these into, again, developmental populations. And I'm cutting out a lot of this for time here. And I'm happy to answer questions about how we did this. We actually found that in the cells that were, their gene expression profile was close to a pro-B cell, that we could separate the patients who would be in CR from the patients who were going to have a 19 negative relapse based on their differentially expressed genes. And if we looked particularly at Ikaros in these cells, again, we saw that on the gene level that there was lower levels of Ikaros in both the 19 negative pre-CAR sample, and then that was persistent in the 19 loss later time point. But of course, this wasn't seen in the patients who were going to go into a CR. So this became a candidate for us to go after and understand how could Ikaros be regulating CD19, which is not something that's been previously described. And I mentioned that, we know Ikaros is implicated in relapse for ALL. It's crucial for healthy lymphopoiesis. And is important both in BCR-ABL ALL and also PH-like ALL, and of course has been found to have germline mutations as a risk factor for ALL development. So Pablo went and started to knock down Ikaros through various methods in a suite of ALL cell lines. On the left here, this is with lenalidomide degradation, and we could see with decreasing Ikaros, you see decreasing CD19. The same happens if you use shRNA. ALL cells don't like to not have Ikaros. We couldn't actually completely CRISPR it out because the cells just die. But certainly shRNA showed us that there was, a relationship between Ikaros and CD19. So Pablo went to his, with these cell lines and started to do some additional omics, so RNA sequencing and then attack sequencing to understand, what is the effect on CD19 transcription or accessibility. Just looking at CD19 expression based on all of the different transcripts, we didn't see a difference in the CD19 level of transcripts, but we did see that if you looked globally, that the knockdown cells, the Ikaros knockdown cells, had certainly a difference in their transcriptional profile, as shown here in this heat map between the knockdown cells in red and the wild type cells in blue. When we looked at the differential gene programs that were enriched after knockdown, we found not only some ones that may have been expected, but we were really interested in things like mRNA processing and RNA splicing, given what we know about these splicing changes that have already been described. And so when we went and looked at CD19, the different splicing transcripts, what we found was that after Icarus knockdown, there was a more of CD19 transcripts were retaining intron 10. And that was a variant, the splice variant that hadn't previously been described in this situation. And so Pablo went and did some more work to look at this. There is an isoform of CD19 that retains intron 10 that also tends to retain intron 2 as well. So we went and looked at other data sets of CD19 targeted therapy, both large cell lymphoma that used a CD19 28Z CAR T cell. So there's 2 data sets that we were able to get access to. And then also another data back to Charles and Catherine's data set in Blina, where we could look at, for the samples that we had pre and post 19 negative relapse, and there was RNA sequencing data, which altogether was about 14 samples. And we found this basically the same changes in the CD19 transcripts, where they had retention of intron 2 and retention of intron 10 more frequently at relapse compared to the screening time point. And this is now across different CD19 therapies. So we think that this is a mechanism that may be part of the regulation of CD19 that is not necessarily specific to a CD19 4-1BB CAR. We hypothesize that this is actually due to changes in RNA binding protein that may be mediated by in the Icarus knockdown condition. There have been, we found basically 12 RNA binding proteins that are differentially expressed between the 2 conditions. And we have experiments ongoing right now to mechanistically look at this and see actually if we are able to detect these RNA binding proteins on the CD19 mRNA, but these are binding proteins that are predicted to be associated with the intron 2 and intron 10 area of the CD19 mRNA. When we looked at our attack-seq data, then we also found that Icarus, as we would expect, also changes accessibility to key lineage transcription factors and their binding sites, because we know Icarus plays a role in chromatin state and open and closed chromatin. And so in the knockdown condition, what we found was that the chromatin state was more similar to a common lymphoid progenitor or myeloid type cells than it was to cells that were committed to the B cell lineage. And when we look at the specific transcription factors that this seems to affect, we see things like BCL11A and B, SPY1, which is PU1, which is of course sort of a myeloid transcription factor, EBF1 and ELF2, which these are all of course sort of transcription factors that are making lineage decisions sort of earlier than a pro B cell population. And this correlates of course with differential expression of transcription factors in general in these Icarus knockdown cells. And if we went back to our single cell RNA sequencing data, we can see that in the patients who are going to have a CD19 negative relapse, their gene expression profile is enriched for an HSC multipotent progenitor state compared to the patients who are not going to have the 19 loss. So we think that it's 2 things. There's sort of Icarus is both sort of changing the lineage or the lineage let's say tilt or lean of these cells, that they may be a little bit more plastic in their ability to look more myeloid, and also specifically changing how CD19 is spliced. When we worked together with Maria Caterina Roti and the Maisner lab to see what happens when we expose our Ikaros knockdown cells to CD19 41BB CAR T cells, we could see, of course, that their CAR T cells make less IL-2 and interferon gamma in the knockdown when they're faced with the knockdown cells. And they also did not kill as well. And this is all in vivo work. And we have the mouse experiments ongoing right now to see if we can recapitulate this in vivo. And I will say that in vitro, we see the same thing if we use blinatumomab and CD19, CD28 CARs as well. So this, again, seems to be something that affects the ability of the CAR to function as well. So this leaves us with a model that looks like this, and I'm sort of wrapping up this part of this story, where we think that before a patient gets a CAR T cell, of course, they have a diversity of cells there. For some patients, they may have cells that already have this sort of lower level of Ikaros transcription factor. So we don't know if we don't believe that these are mutated, but from the samples that we have from the primary samples, the patients that we know of did not have Ikaros mutation. So that's an open question. What does mutation do? But at least they have a low level of the transcription factor. Perhaps then that actually influences the expression of RNA binding proteins, which lead to alternative splicing, and in this case, intron 2 and 10 co-retention that leads to loss of CD19 protein and then a 19 negative relapse. So now if we turn to sort of a question that is where we started with this whole thing is, what about relapse after upfront chemotherapy, which is, of course, the one we really want to try to prevent from happening. And this is published work that I'm just going to touch on to sort of set the stage for the second story I want to tell, which is we looked at 60 primary samples at the time of diagnosis from various subgroups of genetic subgroups of ALL, and looked at them by CyTOF after ex vivo stimulation, which helps us look at signaling changes in these cells. And to summarize this paper, which we published about 5 years ago, what we did is take the single cell data, applied a machine learning model to try to find the cell populations and their features that are associated with relapse. That identified for us basically 6 predictive signaling features that were associated with future relapse, because again, this is the diagnostic sample that we're looking at. And they implicated sort of active signaling, basally active signaling in the mTOR pathway for patients that were going to relapse. And when we use this model then to basically make relapse predictions and compare to how it worked either with NCI criteria or MRD criteria, we could see that if we combined it, basically, which is the solid lines with NCI criteria or MRD criteria, that we could improve relapse prediction compared to just using sort of the clinical tests that we use alone. And so this left us with this model where we call these relapse predictive cells, that at the time of diagnosis, you have billions of cells in there, like I mentioned, and there are some hidden cells that potentially have this signaling network. In particular, these cells are right at the time that these cells are kind of transitioning from a pro-B to pre-B cell, and that these are the cells that then can mediate relapse. So kind of are these the queen bee cells? And now I want to talk a little bit about this metabolism story that we're getting into and chemotherapy resistance. So we've been thinking, about how signaling results in metabolic changes in these cells that could result in chemotherapy resistance. So essentially, I'm going to start this story by going way, way back to the time that I was in Gary Nolan's lab, and we actually did spend some time looking at the gene expression in normal B cell progenitors. So after we had published this B cell developmental paper, I was very interested in, what are the gene expression changes that sort of happen as these cells are developing. And this was before, there was really accessible single cell RNA sequencing. So we had done some sorting where we actually FACS sort healthy bone marrow. We actually came up with a way to do it using TDT to do an intracellular sort, so we could get the same populations that we knew kind of were spanning that expression of TDT. And when we looked at the changes in, again, gene pathways that were happening across development, what was really interesting was, of course, we expected some of these B cell receptor signaling, IL-7 signaling, PI3 kinase signaling, but what came up was glucocorticoid receptor signaling. And that was really interesting and unexpected, because there hasn't been anything or much in the literature about the role of glucocorticoids in the development of early B cells. So we went and looked at this on the protein level again in 3 more healthy donors. And indeed, we could see the glucocorticoid receptor is most highly expressed at the pro-B stage in healthy cells. And this is at the same time these cells are expressing things like PU1, TDT, CD179B, and like just as they're starting to turn on CD19. So of course, we know glucocorticoids are really important to regulate gene expression, and they also play a role in restricting glucose. And in ALL, they've been used for decades, especially in the European trials, as a way, kind of as like a poor man's MRD, where you could look and see how much leukemia was left after a week of prednisone, and that actually stratified patients in relapse risk. And so we said, okay, this is really interesting. We're seeing this on healthy cells. What happens in leukemia cells now when we look? And so we went to 19 primary samples, some of which were paired diagnosis relapse samples, and we subjected them to treatment with dexamethasone for 48 hours in vitro, and also added dasatinib, and I'll talk a little bit about why. And again, what we found was that it was in the leukemia cells, the pro-B cells are the most highly expressing the glucocorticoid receptor. But interestingly, we could see that in the patients who had less cell death in vitro after 48 hours of dexamethasone, that it was actually the non-responders who had the highest level of glucocorticoid receptor. So it's like the cells that survived had this very high level of GCR compared to the cells that died, and to the patients who their cells were mostly dead after 2 days of dex. We used our developmental classifier here, and we could see that the cells that survived after 2 days of dexamethasone actually looked more mature. So almost like those surviving cells were pushed a little bit in their protein expression to look more mature than the cells that we started with, because this is basically a fold change between the untreated and the dexamethasone treated cells. And when we looked at their intracellular events, we found that there was signaling that was similar to what we had previously described in our relapse predictive cells, that they had activation of CREB, they had activation of S6 after dexamethasone. And when we added dasatinib, which targets Src kinases, which are downstream of the pre-B cell receptor, that we could we could decrease that signaling. And we also saw basically in the poor responding patients that there was sort of activation of glucocorticoid target, and this is looking at the protein level compared to the patients who were good responders. So it seems like these cells are trying to suppress suppress this pathway, and they potentially are overexpressing the glucocorticoid receptor as a way to do this, but they're incapable of it. And to kind of summarize this part quickly and move through, essentially when we either in vitro or in vivo add dasatinib to dexamethasone, we could get improved cell killing. And this also was associated at later time points with decrease in the signaling, especially in S6 and CREB, when we added dasatinib to dexamethasone. And we looked at this actually in primary samples who were treated with an 8-day window of prednisone from one of the AIEOP protocols. And again, we saw similar to what we saw in the in vitro experiments, that after 8 days of prednisone, the cells that remained looked more mature than the cells that started. And again, that there were both phenotypic changes and some signaling changes at that day 8 time point, again, with activation of S6 and ERK, as well as CREB. So what was a really interesting other result that was sort of dovetailing with another project in the lab, and I want to mention all this work was led by Yolanda Sarno in the lab, who just has started her own lab at the University of Milan, and is a wonderful collaborator and researcher, that when we did gene expression, again, comparing dexamethasone survivors versus dexamethasone, sensitive cells, what we found was that there was activation in the surviving cells of signaling pathways like mTORC and pKa phosphorylation of Krebs and mTOR signaling. But we also saw a regulation of metabolic genes and fatty acid metabolism and some other metabolic pathways that was really interesting, because at the same time we were working on specifically following up on these relapse predictive cells. And this all was led by Yuxuan Liu in the lab. And I'm showing her here with her adorable son, Leo, who's a little bit bigger than this now. So we were going back and thinking about our cells that we identified and that they have this particular signaling signature. And in this case, we had also seen this signaling pop up in our glucocorticoid resistant cells. So we hypothesized that perhaps this is signaling that's coming downstream of the pre-B cell receptor, because that's about the time developmentally that these cells exist. They express pre-B cell receptor proteins, and this is sort of a normal developmental checkpoint for ALL cells, B cells to, move on to further differentiation. And Marcus Mushin's group has previously categorized ALL cases into pre-BCR positive and pre-BCR negative based on their signaling, and tends to be enriched in cases that actually have PBX1 rearrangements, but also can be defined in other subtypes of ALL. And so we were wondering if our cells were actually having, pre-BCR signaling. And Yuxuan, we started with this she went back to our original cohort of 60 patients, and we took 6 patients, 3 of whom who would relapse, 3 of whom who would not. Again, this is at the diagnostic time point, and did RNA sequencing on the sorted pre-B cells. And again, looked at differential gene expression. And when we looked at the pathways that were enriched in the patients who were going to relapse, again, we saw the signaling pathways that we expected based on CyTOF, so mTORC, PI3 kinase mTOR. But was quite interesting is we also saw metabolic pathways like oxidative phosphorylation, glycolysis, and fatty acid metabolism. So that sort of pulled us into a metabolism space that was really interesting. And so Yuxuan went and looked at data from the DeepMap portal, which is a fantastic cancer dependency data set where there's CRISPR knockout and you can look at dependency of different genes. And when we look at pre-BCR positive versus pre-BCR negative cell lines, which are categorized based on the gene signature Marcus Mushin described, we could see that in particular, the pre-BCR positive lines were dependent both on the pre-BCR related genes, but also on glycolysis related genes, more so than pre-BCR negative lines. And if you go to the target data set and look at event-free survival, again, based on patients with this gene signature, we can see that, that the pre-BCR high patients have, a worse outcome probability. So we started to get into actual metabolism studies where we did some seahorse in pre-BCR positive and negative lines, and we could see that the pre-BCR positive lines were much more energetic than the negative lines, just overall, both using glycolysis and oxidative phosphorylation. And when we actually did ICP-MS metabolomics, we could see particular pathways enriched in pre-BCR positive cells compared to negative cells. And in particular, again, unsaturated fatty acids, pyrimidine, and purine metabolism were some of the top pathways that were coming up, which was quite interesting. And we did this with Maya Kozlowski here in pathology. Yuxuan then wanted to understand what happens as we modulate the signaling of the pre-BCR, because of course that's how we started down this path, is by trying to look at these active signaling cells. And here's an example of some of this data from the cell lines, that if we activate the pre-BCR by cross-linking it with IgM in Kasumi2s, which are a pre-BCR positive line, we can see that they have increased glycolysis, which is measured by extracellular acidification, compared to REH, which are pre-BCR negative cells. And similarly here, if we inhibit it by using a SYK inhibitor, and the same thing was recapitulated with a SYK knockout, you could see that there was decreased glycolysis in the pre-BCR positive cells only. So there's something specific about cells that have this pre-BCR signature in their metabolism that is different than the pre-BCR negative cells. And then we did some pretty simple starvation experiments where we starved pre-BCR positive or negative cells of either glucose or glutamine for 48 hours. And we could see in particular, and this is summarized here, that the pre-BCR positive cells were much more sensitive to glucose starvation with cell death compared to the pre-BCR positive, or sorry, negative cells, which were not so affected. And none of the lines actually were particularly sensitive to glutamine starvation. So to summarize some of these points, we found that these pre-BCR positive cells are highly energetic with more glycolysis than pre-BCR negative cells. That if we cross-link their pre-BCR, it increases glycolysis. And if we inhibit that signaling, it decreases glycolysis, and they're sensitive to glucose starvation. So that left us with this question, why are they so glucose hungry? So Yuxuan set up a pretty elaborate and complicated experiment with these pre-BCR high and low cell lines, either in a regular media or with glucose deprivation or glutamine deprivation. And then we did carbon tracing at various time points of either labeled glutamine or labeled glucose. And I'm going to summarize the results of this on the next slide here. Basically, what we found was that all the ALL cells, regardless of pre-BCR status, primarily use glutamine to fuel the TCA cycle, and that's indicated here. These pie charts indicate how much labeled carbon and from what source is it going. And so, for the most part, all the cells use it in the TCA cycle, and also for aspartate production. But in particular, the pre-BCR positive cells are using glucose to go through glycolysis and the pentose phosphate pathway. And the pentose phosphate pathway, if I remind you, is kind of the upstream pathway to nucleotide production. And in particular, what we found was that the pre-BCR high cells are really fueling pyrimidine metabolism. And so that's the uridine and the adenine and thymidine for DNA. And so this is some of those products in the pre-BCR positive cells compared to pre-BCR negative cells. So always higher in the pre-BCR positive cells. And in fact, giving back uridine, which is the RNA particular nucleoside that is a pyrimidine, and that's shown here in this sort of maroon color, can rescue pre-BCR positive cells from glucose starvation. And so, but not necessarily just a pooled pyrimidine that has the adenine and the thymine. So suggesting perhaps that these cells are needing the uridine for RNA production, although that's ongoing work that we're trying to understand. We know that pyrimidines serve a lot of roles in cell growth. So if I kind of focus in over here, this is the pathway that they use. And if we zoom in even more, to make pyrimidines, you need to start with glutamine, and then you have to add aspartate. And then eventually there's a rate limiting enzyme step that happens in the mitochondria called DHODH, which is dihydroorotate dehydrogenase. And eventually then you go down to the actual pyrimidine synthesis pathway. Pyrimidine metabolism now is emerging as sort of a really interesting, I think, area in cancer. So it's been identified now in IDH mutant gliomas, as well as diffuse midline glioma and pancreatic cancer. These papers are all in the last couple of years. And in all of these cases, the cells are using pyrimidines for different things, which is interesting. And so that's some of the next steps that we're working on in our work. But then we wanted to go back and say, okay, how does this connect to the signaling? Because we started down this path of metabolism by, identifying these signaling cells. And going back to some, previous literature that PI3 kinase signaling, or mTOR signaling in particular, as well as MAP kinase signaling, actually activates or stimulates de novo pyrimidine metabolism. And it happens by phosphorylation of the enzyme CAD, which is one of the first enzymes in that step. There's actually multiple places where there are activating phosphorylations that can be downstream of these signaling pathways that tell cells to make pyrimidines. you can inhibit pyrimidine synthesis. There's actually a couple clinical trials in both solid and heme malignancies looking at this target, which is dihydrooro. It's a DHODH inhibitor. And we could see that our pre-BCR positive cells, and which is on the left, and PDXs, which are on the right, are more sensitive to this inhibition. If you, this is all in vitro data. And again, we have mouse experiments ongoing to see how this works in vivo. And then we've also developed a metabolism panel in collaboration with Sean Bendel and Felix Hartman, from who are close friends and collaborators here, and Lucille Stuani, to be able to look at expression of some of these metabolic proteins in primary cells, because metabolic studies tend to need millions and millions of cells to do some of the work, like on Seahorse or ICP-MS metabolomics. And that is something that we are working on, looking now at primary samples. And this is an example of enolase 1 and LDH, which of course are, downstream enzymes in glycolysis. And so we can see that these are in 3 primary samples in each column. A diagnosis sample from a patient who won't relapse, a diagnosis sample from a patient who will relapse, and then their paired relapse sample compared to a healthy bone marrow. And each row is a developmentally classified population. And we can see that in the patient who's going to relapse, both at the diagnosis and relapse time point, there's increased expression of enolase and also of LDH in particularly those cells across the pro-B to pre-B transition, as we would expect based on all the data I've showed you to this point. And so now we're expanding this to look at more samples. So coming back to this question, it leaves us with a lot of new questions to explore that we're really interested in working on as we move forward with this work. So we want to understand more, of course, how do the genetic lesions that we know are common in ALL that result in activated mTORC and RAS signaling, do they actually enforce this metabolic phenotype? And does that have to do with why those are may tend to be more relapse prone? Also, PAX5 and Icaros have again been described as glucose regulators by Marcus Mushin's group in B cells and in ALL. So does this have to do with, if we have mutations in those transcription factors, does that mean that these cells have more access to glucose that then they can further use for this pathway? We don't know if this pyrimidine metabolism results in treatment resistance per se, and if targeting this metabolic state is effective, but I think it is a novel target, of course, in ALL. And then, there's a lot of clinical factors that we know are important for relapse in ALL. We know obesity is a risk factor. We know hyperglycemia can be also associated with relapse risk. And of course, we induce metabolic changes in our patients by using glucocorticoids and asparaginase. So I think this really opens up a lot of questions about, what these cells need glucose for, and is this a targetable vulnerability? And so with that, I'm going to end. I have a few minutes left for questions. And I want to thank, of course, all the members of my lab, all of our collaborators, and of course, all of our funders, and of course, Alex's Lemonade Stand for all of their support always. And I'm happy to take questions.
Thank you, Kara, for a wonderful talk. We have one question that was posted in chat, and I can read that to you. Do you think discordant cells are a phenomena specific to post-CAR T relapse, or are discordant cells a feature of relapse in general? Are discordant cells LSC?
I think that's a great question, and something that is definitely a question I would like to explore, but we haven't actually had sort of time or bandwidth yet to do that. But certainly we see them in the upfront samples, as I mentioned, in diagnostic samples. So I think it's a really interesting question, and certainly not only has implications perhaps for CD19 therapies, but plasticity, or whether or not it's possible, or whether or not it has anything to do with like lineage switch cases or patients in which we think are at risk of lineage switch. How do these cells play in those patient groups? So it's a question we would love to answer, and just need the time and the people to do the work.
That was one of the questions. If anyone has questions, you can post it using the Q&A feature or in the chat box. I just have one question or a comment.
Sure.
Are you planning on then, with your work with patients where you're looking at predictive cells for relapse, also extending it to looking at treatment resistance and looking at the single cell
Yeah.
phenotypes for that?
We have an ongoing project. Actually, Yolanda Sarno, it's sort of something that she started here and is taking with her, is looking at MRD cells as well. We have like a pretty amazing data set with about 50 patients with paired diagnosis, day 8, day 15, and sometimes relapse samples that we've looked at by CyTOF, and now are doing some single cell RNA and DNA to then specifically say, are the cells that we know are treatment resistant, like MRD cells, how do they look compared to actual relapse cells and the cells that we predict to be associated with relapse? Again, I think eventually getting to a place where we can, come up with a definition of a leukemic stem cell or features of leukemic stem cells in ALL, because of course it may be diverse. It may not be one size fits all for all patients, but there may be some shared features that we may be able to identify.
Does
And I see
one,
another question.
yeah,
It
go
popped
ahead.
up. Do you envisage incorporation of cell characteristics will be applicable prospectively to predict relapse? I think that would be sort of the holy grail of the work that we're doing, is to be able to do this as a clinical test. One of the other projects that we're working on is to see if there's ways that we can take some of these signatures that we've identified and make them something that could happen in a clinical flow lab. Doing phosphoflow cytometry in a clinical flow lab is not really realistic. So we've been doing some actual surface marker screens to see if there are surface markers that associate with these that then could be integrated into flow panels fairly easily. And then also if we identify some gene signatures that may be associated, then that may be something that's easy to do as a diagnostic.
As a follow-up to that, um, your work focuses on bone marrow as an analyte, right? Is
Yeah.
it possible to do the same with peripheral blood
Sure.
samples?
Yeah, we've done a lot with peripheral blood. And of course, peripheral, and it's not necessarily interchangeable that the populations are exactly the same, but it has to do mostly with whether or not the patient has sufficient leukemia cells in the peripheral blood for us to detect. But even in the MRD project, we can see cells in the peripheral blood, even at about the same sensitivity as flow cytometry, which is what you would expect.